thank you to all of you for taking the time to come along today. My name's James Stevenson, and I lead the development of ProtVar, this resource that will hopefully help you to contextualise the consequence of your human missense variation. I'm joined today as well by Tanushree, who you can still see on screen there, waving. She's one of our data scientists, and then Pravat, who's our lead developer on ProtVar. And they will be looking at your questions, hopefully answering some on the fly, and then questions that are more complex we'll get to at the end. Okay, so I'm talking about contextualizing human missense variation. So perhaps we should just start with some small definitions. I'm sure because you came to this webinar, you probably know most of these, but when we talk about genetic variation, what I mean is sequence differences between individuals of a species, in this case humans, from a reference sequence. There are lots of different types of genetic variations: structural variations, SNPs, single nucleotide polymorphisms, insertions and deletions, copy number variants, translocations and inversions. Specifically, ProtVar is trying to help you contextualize missense variation from single nucleotide polymorphisms. Why that one? Well, by far they're the most common of all variants. Well over 90% of variants are SNPs, and that equates to about 4 million variants per person. And of course, SNPs in protein coding regions, which also change the amino acid sequence, as you can see here on the right, are called missense variants. So as you can see here, this middle position of the codon has changed from a G to a T, and therefore the amino acid has changed from an arginine to a leucine there. And numbers-wise, we can think about maybe 9,000–10,000 missense variants per person. This is 9,000 different positions where your amino acid is different from the reference sequence. Of course, most of those are benign, but some are not, and they're very specific to the position. So a change in one position in one protein will have a very different consequence from another one, which makes the interpretation of missense variation very complex. Which is why we developed this resource called ProtVar, protein variation, to contextualize this variation and help you to interpret the consequence. So what it is a variant-centric collection of relevant annotations rather than being nucleotide-centric. It maps variant formats and versions, which I'll show later, and it retrieves and organizes data from all over to try to help you understand the consequence of your missense variants. We also have some novel predictions that are not available elsewhere, specifically for missense variant interpretation. And ProtVar, since its release almost 2 years ago, is used by many different people in industry, across academia and in health services right around the world. If you want to have a look on your phones, of course, it's not optimized for mobiles, but you can see it using the QR code. Probably better if you use the link on screen. Perhaps you can look along as I'm talking about it, and then we can come up with some questions later. Okay, so mapping really is the core of ProtVar. Without the mapping, it's very difficult to get annotations from different sources. So when I say mapping, we're talking about mapping between different formats, different versions, and of course also mapping between the chemical languages of biology. So between nucleotide and amino acid sequence. So you can see on this example on screen, ProtVar helps to map between different kind of IDs, cosmic IDs you can see there, and dbSNP IDs, different formats as far as the nucleotide coordinates, VCF formats or HGVS formats, also cDNA formats there as well. And you can see going to the right, we map everything to GSH38 coordinates and then to protein positions, different isoforms, alpha structures, and PDB structures as well. So ProtVar itself, the tool, accepts all of these different formats and then standardizes it, maps it all onto a common format in order to help you navigate the different annotations. We have high coverage, not for every single protein, but for now over 93% of human proteins, we have mappings, and we also allow mixed input. So if you happen to have some GSH37 and 38 coordinates, perhaps you have some IDs and cDNA, you can enter all of these on this next page. This is what ProtVar looks like when you first go to it. This is the home page. You can see there's a paste box, and in that paste box you can enter your genomic coordinates, cDNA, protein, variant IDs, or any mixture of those. The other thing ProtVar will do is it will convert between GSH37 and 38 for you, even if you don't know whether your variants are in 37 or 38 assembly. ProtVar will actually detect that for you. If you paste a load of variants that are in GSH37, ProtVar will check against the 38 sequence to see whether you have 37 or 38. And if you've pasted 37, even if you didn't realize that's what you had, it will convert them to GSH38 for you and then submit them without you even seeing the conversion. Also, whole proteins. So you can see at the bottom here in this green box, you can add a uniprot accession. So you can when you add a uniprot accession, it will bring up all the possible variants in that protein and all the annotations for all of the variants. So if you have a favorite protein that you're working on, simply enter it in the box at the bottom, and then it will bring everything we know about that protein all to you. So this is the next screen after you've pressed submit with your variants. There's no limit to the number of variants you can add. You can look at one variant, two variants. You could paste 500 or even 1 million variants into that box, and this is still the screen that you would get afterwards. The mapping is very, very quick because we have pre-mapped all of those different formats and versions. So you'll get this next page almost instantaneously. So we'll also show you a summary of the transformation. For example, if we've changed from 37 to 38 formats, it will explain that to you. And if you've got mixed formats, it will keep a track of which kind of formats you have added in the output screen here. But you can see here, this has been standardized and mapped to the protein positions. So in this case, we've entered some genomic coordinates on the left. You can see the reference and alternate nucleotide that you've added. It maps it to the gene, the position within a codon, a CAD score, which is a nucleotide-specific score of variant effect. The isoform here, I will show you later when I show you a live demonstration, but you can actually have a drop down here where you can see the position in all the isoforms and not just the canonical isoform, the protein name of your variant, the position within the protein in the canonical isoform, the change, the consequence, the alpha missense score, and then really importantly, on the right here, we have 3 different buttons that can be clicked. And these 3 different buttons store all of the annotations for the variants. The first one, functional annotations, everything we know about the function of that particular residue in the protein. The next one, the population observations, everything we know from the human population about that position. And then finally, the structural information. So this stores the experimental and AlphaFold structures, any ligands that are mapped to it, so that you can see your variant in the context of structure. I think the key word about this page is standardization. So no matter what kind of variant format you add, you will always get this sort of output. And following on from that, all of the different scores we have, we've also standardized the scoring and the coloring and categorized them. And you can see here in the legends. Whereas some scores go from green to red, blue to red, we've standardized everything so that in ProtVar you'll always see a benign variant in a kind of blue color, a more pathogenic variant in red colors. Okay, so clicking on the first button, this one looks like beads on a string. These are functional annotations, and they're split on this page into 3 different parts. The first part shows annotations that we've brought from UniProt, which are specific to the position, to the amino acid position. So you can see in this particular example that the position from histidine to arginine has some literature which suggests it's an active site proton donor acceptor. There's a paper that shows that there's a 3,000-fold reduction in activity when you change from histidine to arginine in this position. and yet another paper which shows an even greater 15,000-fold reduction in activity when you make this change. If you click on these, you can see the PubMed IDs, and you can go to the original literature where it shows that. You can see below there are scores. So conservation is something that's directly calculated by ProtVar, but you can see also there's a predicted stability change, which I'll go into later. This shows how likely this histidine to arginine change in this position is to destabilize the overall folding of the protein. And then we can see the pathogenicity predictions. The CAD score, which I mentioned earlier, alpha missense score, calculated by DeepMind, and the EVE and ESM1B scores as well. These are all measures of pathogenicity from third parties that we bring together. And we have, again, all of these stored and pre-calculated, including over 200 million stability changes that we calculated. All, of course, standardized by our color scheme. If you click this, though, you can get the original color schemes from the original publication if that's what you prefer. So on the right, you can see more annotations, but these annotations are the region that the variant is in, rather than just the specific amino acid itself. So here you can see that it's in this particular chain from UniProt, and then we can see some predictions. These are predictions from ProtVar. The first one being that this variant is in a pocket. It's in a protein pocket, so a potential small molecule binding pocket. And you can see a confidence score here, and you can see all of the residues that are involved in that particular pocket. And later I can show you that pocket in the context of a structure. And then secondly, we can see that the variant is in a protein-protein interface, or rather we predict it to be in protein-protein interface. And then we also have the P.Q score, which is a confidence score, how confident we are that that protein-protein interface is at those residues. So this isn't an exhaustive list by any stretch. This shows you some of the sources that we get our functional data from. And for sure, the most important one is UniProt, because UniProt already collates functional data from so many different places. I've just lumped it all into EMBL-EBI because UniProt collates protein functional information from so many different resources within EBI and then standardizes them, maps them to the protein position. So we use all of that information. But the other incredible thing UniProt does is with their team of curators, they take experiments from literature, directly from literature, curate it to check validity, and then add it to their entries. So these are the two major sources. So these kind of high throughput data from other EBI resources, and then also curated, highly curated, expertly curated data that goes into UniProt and then into ProtVar. The protein-protein interfaces and the pocket things that we showed, this was via a collaboration with Open Targets using AlphaFold structures, which I'll show you later. And then the scores come from many different places. So three of the examples are the CAD scores, the POP-EVE scores, which have been integrated extremely recently, and then the DeepMind alpha missense scores. Okay, so moving to the second little button, the second button with annotations for your variant. So this is still the same variant as I showed before, or an equivalent variant, and you can see here that the left-hand side shows all of the different resources that report that variant. So this variant's been reported in ClinGen and ClinVar, UniProt, Ensemble, and dbSNP. And you can see it's being categorized in ClinVar and in UniProt and Ensemble as pathogenic. So this is from those individual resources. And if you click on any of these, you will go to the original entry for all of those resources. On the right, we have something that's quite different from other resources, which is co-located variants at the residue level. So here we mean other changes to the reference alanine. This is not necessarily at the same nucleotide position. It may be another nucleotide within the codon, but it is changing the amino acid. And the reason that could be important is that perhaps your variant, which you've uploaded, has never been seen before in any of the resources on the left. And we don't really know what it does. But at the same amino acid, this alanine, for example, perhaps that has been seen in humans. And changing the alanine to something else may lead to a disease. You can see the disease listed here. So this is a way of expanding our knowledge about this reference amino acid by considering literature and resources that have shown collocated variation. So again, many places we get the variants from. Nomads, we get not just variants, but also some allele frequencies. This symbol here is cosmic. So this is where we get our cancer-specific data from. ClinGen, not just variants from here, but also disease associations from ClinGen. We get lots of our data from Ensemble. UniProt, as always, is a huge source for us. And then dbSNP from the NCBI. Also, we retrieve lots of variants. Okay, so I think one of the strengths of ProtVar is it's not just an aggregator of scores. We don't just show you the alpha missense scores and the CAD scores. The scores help you understand whether a variant is likely to be pathogenic or benign. What ProtVar also tries to show you is how the variant is pathogenic. What is the mechanism? What's the molecular mechanism of the action? And the way I look at this is broadly split into two different options. Either a variant can alter folding, and altering folding can be many different things. This could be, breaking important bonds, steric clashes, where you put a large side chain instead of a small side chain, or the opposite way around, where you get a large side chain and you swap with a small side chain and create a void. It can change the torsional angle. For example, glycine and proline are on complete opposite ends of the spectrum as far as the permitted torsional angles. We can think about gain and loss of charge or hydrophobicity, which alters folding as well. So there's many different ways that amino acid change, a single nucleotide variant missense change might change folding. But it can also directly alter the function of that amino acid, which of course might lead to disease. So one example may be that it changes the active site in an enzyme. This can be catastrophic, of course. Active sites are extremely well conserved, so a missense change there could have a huge effect on the protein function. But it may also interfere with ligand nucleic acid binding. So ligand being small molecules binding to pockets, nucleic acids, RNA and DNA, that might bind along a groove. This might not just be reducing the affinity of a ligand, it might be changing specificity, it might even be increasing the bond between the ligand and the protein, which may have an equally bad effect on the organism as a whole. Protein-protein interfaces. So amino acids along the protein-protein interface that are changed may again affect the dynamics between the two proteins and either bind too strongly or not strongly enough and then affect the downstream signaling. And finally, disrupting post-translational modifications. So swapping amino acid may not allow the same modifications to occur, and that may have a biological effect on the protein. And ProtVar doesn't look at all of these, but most of them. And actually, we're working on all of the ones that we're missing. The PTMs, we have an active collaboration at the moment with the Functional Genomics Initiative in the UK in order to understand better how variants can affect PTMs, which will then be added to ProtVar. So let's start with some of the predictions that we do have. I mentioned protein stability earlier. So this is where a single nucleotide change causing a missense variant then significantly changes the stability of a protein. So we predicted this using FoldX version 5 on AlphaFold 2 monomers in regions that are well predicted by AlphaFold. So this is not the N terminals, which in some cases are spaghetti-like and not very specific. This is more of the core regions. But even so, we're talking about over 200 million individual predictions, which are all stored in ProtVar and therefore instantly accessible, either variant by variant, and we also offer them as a download if you want all of our variants all at once, all our stability predictions, then you can take that. So you can see an example there, there's threonine to phenylalanine, and you can see there's the green one is the reference, and then you see the variant, and then the induced change is categorized in terms of destabilizing or not destabilizing, and then offered in ProtVar. This is a collaboration with Open Targets, and the paper here you can see, and you can go and have a look at the bioRxiv paper before publication to look at how we did these things. So the second really kind of new prediction that's in ProtVar that I think is useful for contextualizing and interpreting missense variation is our protein-protein interactions. So we tested 500,000 different protein-protein interactions that have been shown either experimentally or predicted as pairs, and of those 134,000 passed our quite robust filtering of models that we were happy with. The confidence was high. So you can see these 134,000 models are all available in ProtVar. And if your variant, so the variant is in yellow here, is at an interface, you will get a model like this. You can download the entire model and then you can play with it in your own browser. Or you can just see where it is in the structure. We also have a confidence score. So even though we're happy with all these 134,000 models, that they have high enough confidence to display, some are more confident than others. In many cases, a variant will have 3, 4, 5, 6 different binding partners at that interface. So we show all of them, but with a confidence score. So you're able to rank which of the interactions is more likely to be important in interpreting your variant. And again, that's in the same paper, which I encourage you to have a look at. And then finally, the protein pockets. So these are regions of proteins that have the right geometry and the right chemistry to suggest that a small molecule might bind to that region. And we've predicted this using AutoCite based on AlphaFold 2 models again, and we have over 100,000 of these refined pockets. I say refined because AutoCite tends to over predict pockets, which means that a protein like this might have 4, 5, 6, 7 pockets predicted. But what we've done is we've refined down to the most likely pocket for that protein. And again, we have pocket confidence for everyone, so you can filter just the pockets which are most likely to be real. We also create some feature points which could be useful in the future for lead optimization for the kind of small molecules that bind into the pocket. And in that same vein, we also use alpha fill structures to show in ProtVar. This isn't our creation, this is a third-party alpha fill. But what alpha fill does, it aligns the AlphaFold structure with experimental structures which have ligands. And once the backbones are aligned, the experimental structure is removed, leaving the ligand in approximately the correct position in the AlphaFold structure. So you can see in this example, again, we have the variant in yellow, and you can see a ligand which has been put in approximately the right place from the experimental structure. And you can see in this case that the ligand is very close to the variant. Equally importantly, you can see 1 or perhaps 2 ligands in the region of the pocket, which gives us more confidence that the pocket has been correctly predicted. So all of those structural annotations can all be seen in the third of the 3 buttons, the structural annotations button. So this, as you can see on the right here, this lists all of the experimental structures in a table. These are experimental structures which feature the variant position. These are not all of the experimental structures for the protein, but they are all of the structures that have that position in them. In the second table, you can see is the predicted structures, these are the AlphaFold structures. The second one is the AlphaFill structure, so that's the structures with the ligands bound. And then finally, which I can show you in the demonstration, we have the predictions of the protein-protein interfaces, which are all custom models, which again are downloadable, and you can take the custom models from us and you can use them yourselves. Okay, so perhaps you've entered 1,000 variants or 10,000 variants into ProtVar, and you want to share them with a colleague and show the analysis that you've done in ProtVar. Well, we can do that. On the left side of the bar, perhaps some of you are on ProtVar and you can see this. There's a search history. you can see all of the searches you've had. And by clicking on the search history, you have a table like you see here. And within the table, you have the sharing links. So you can click this and it copies a URL to the clipboard. You can share that with your colleague. And when they enter that, they will immediately populate ProtVar with all the variants that you have submitted. And as well, your downloads, which I will go into, but all the information that you've got in ProtVar, you can download it all into a CSV so that you can filter it and play with it yourself. Sometimes these downloads, if you've submitted many variants, they can take a while to create, but that's okay because you can share them with your colleagues here and you can download them onto your local computer here as well. And again, it keeps a list of all of your downloads that you've created. If you're running into trouble in ProtVar and you don't quite understand how something is being defined, for example, then we have a large help section. So the help section, you can look at the different categories, but also all the way through ProtVar, you see these little eyes with a circle around it. And by clicking on those, it will bring some help in from the side and it will explain the section that you're looking at to help you understand how to use ProtVar. And of course, you can always contact us as well at protvar@ebi.ac.uk with any questions or suggestions. So as far as accessing it, I've already been talking about the UI, which is a brilliant way to explore and manipulate the data in ProtVar. But there are other ways to look at our data. If you want to, for example, link directly to ProtVar or look at single variants or single proteins or single nucleotide positions, you can very easily use this simple URL structure. So for example, here we've got the G to C change in this position in chromosome 19. By entering this will go directly to that result in ProtVar. Equally so as a protein, so it's an S to R change. You don't know what nucleotide change it is, but that doesn't matter. You can just go to the accession, the position, and the change, and it will show you all the results we know about this. And then if you have an RS ID, a DB SNP ID, for example, or a Cosmic ID or a ClinVar ID, again, enter that and you go directly to the result. If you want to download, for example, all of our predictions, we have an FTP site. So I showed you the pockets, I've showed you the interfaces, I've showed you stability. This FTP will allow you just to download those predictions that you may want to embed into your own work. Programmatically, you can use our API endpoints. We have many different API endpoints, so you can get straight to the information that you want and you can embed it into your code. And then finally, the download. The download is either everything you see on screen, or you can be much more refined and filtered and just look for the structural annotations, the functional annotations, or perhaps you just want the mapping. Perhaps all you want is going from some IDs and some messy inputs, which have 37, 38 cDNAs, and you just want the GSH38 mappings back. You can do that as well, and it's very fast, and you can retrieve the download of tens of thousands, hundreds of thousands of your messy variants, all standardized by ProtVar. So just to spend a few more minutes on what the programmatic access looks like. Of course, I don't know who's here as far as how many people use APIs and understand the utility of them, but I would encourage you to go and look at the API section of ProtVar. It's quite clear, and it will run you through how to use an API and why it's particularly useful. So in this case, there's a score endpoint, and you can see I've just entered an accession, a position, and a mutated residue. If I press execute, it will create a URL for me. It will also show me all of the scores because I'm at the score endpoint. This just shows me the Eve scores, alpha missense scores, conservation scores, ESM scores for this position and this mutated residue. So we can make this as complex as we want to, and we get more information back responded from the API. The download, I mean, I didn't want to show you the many columns in the download because I can't think of anything more boring than looking at an Excel sheet. But you can see an example here. So you can see how many columns there are. And basically everything that we have in the UI you can get in the download. Of course, that will then help you to filter and sort all information, especially if you've entered lots and lots of variants and you just want variants with a specific criteria. The download is very useful. And as I say, you can generate this entire table, or you can just generate the parts that you want, the functional annotations, population, observations, or structure. We update ProtVar often. This is the external, all of the scores we update quite often. Every time there's a new major update, for example, CAD recently updated from 1.6 to 1.7, we very quickly added that. I would say also that all of the UniProt annotations are updated continually, and that's because we use their API in order to retrieve the information. So all of the functional information in ProtVar is updated, more than daily. So the mapping itself is quarterly, but actually all the information in function is all the time. So you'll always get the most up-to-date information from UniProt via ProtVar. So hopefully I've tried to show you and help you to understand that all of these different aspects of missense interpretation, they're interlinked. They're all very important. And we try in ProtVar to bring them all together, not to tell you the answer, but to help you to interpret and contextualize your information. So you can see here how pathogenicity predictions clearly are linked to human conservation and to function, but also they help in variant mechanism, and eventually they help in understanding disease. And ProtVar sits in the middle of this, helping you to understand how your single variant can link all the way through to variant mechanisms and to disease. So I hope to show you. Okay, so I just wanted to show you, briefly what it looks like. So here's the homepage, which I showed you before, and here's this huge box, which I can type my variants into. If you don't have any variants to hand, you can use these examples. I'd encourage you online maybe to open the link. It's ebi.ac.uk/ProtVar. You can maybe do this along with me so you can see how to use it. So let's just look at the genomic coordinates. You can see the genomic coordinates here have all different formats. So you can see there are some HGVS formats. There's some VCF formats, but different types of VCF formats, sometimes with dashes in them, spaces in them, forward slashes. That's fine. ProtVar can interpret these in most cases. It's not trying to trick you. We're trying to be as helpful as possible to our users. You can see here also there's an auto detect, which is clicked. If you know that your variants are GSH37, then of course you should click this. But if not, we will predict it for you and then change any 37 to 38 if needed. So from here, we can press submit. As I said before, this takes you very quickly to the results table. The results table explains to you that you've entered 4 genomic inputs. If you had mixed inputs, it would then list out genomic inputs, protein inputs, cDNA, etc. It also shows that we've predicted the inputs to be GSH38. All of them match 38 in this case, and none of them match 37. Okay, so we can go across here and we can see all of the different standardized data that I mentioned. But what we didn't see before is the drop down here. So in this case, you can see the different isoforms. In most cases, the position is the same. But as you can see here, in this particular isoform, the amino acid that you've entered is in a totally different place. So position 134 rather than 493. So this may be relevant to you. So clicking on these 3 buttons, these are the 3 key buttons. The first one is functional annotations. So again, we can click on these things and we can see the PubMed ID that mentions the data above. We can see all of the predictions down here. Again, by pressing prediction, you can get the help that comes out from the side. And here we can see the pockets with the confidence. We can either show all of the pockets, or we can just show the high confidence, very high confidence, low confidence pockets here. And then again, here we have the protein interfaces, which shows the confidence here as well. So on the next tab, we can see, which I didn't show you in the presentation, that we have the exome allele frequencies from Nomad. If you click on here, it takes us to the Nomad page that shows that. Hovering over this tells you the exact allele frequency, but we've categorized them. So this is a very rare variant. It shows the other resources which have reported this variant. And by clicking on these, it will take you to external sites of the original source. And then on the right here, it shows you the variant that is co-located in the same amino acid. And again, this one is cosmic. This is a cancer variant. And by clicking on the cosmic link, it will take you to the cosmic page, which will describe more about this variant, which is co-located at the same position. And then finally, by clicking on the third button, it will show you all the structural information. So this viewer is a Molstar viewer. We can make it full screen, and there's many options we can select here, including having it rock and rotate. These are all of the experimental structures, and you can see in this case there are many experimental structures which have our variant position in them. We can zoom to the variants by using these buttons at the bottom and highlight the different chains. And if we go to the next table down, we can see the alpha fold structures, colored according to confidence, and then below that the alpha fill structure. If you remember, alpha fill has the residues, the ligands in approximately the right place on the alpha fold structures from the experimental structure. Again, we can zoom to the variants. We can highlight the pockets. So in this case, you can see the pocket highlighted in magenta. And then we can zoom into the variant position in yellow, and you can see the ligands that are nearby the variant position. So I encourage you to play around with this. A ligand's near to a variant is one of the surest mechanisms that we have. And then finally, you can see the protein-protein interfaces in the third of the tables. So these models, these are not AlphaFold 2 models. These are created using AlphaFold multimer, which means both of these chains have been they've both been modeled in each other's presence. They're not modeled on their own. So these two chains have been modeled together using AlphaFold multimer. This is a custom model, which is downloadable from ProtVar. And you can see here where your variant is in yellow, and you can see the two chain. So clicking on highlight interface shows you all of the residues that are at the interface and also in the shell just behind the interface, because often variants that are in the shell behind the interface can interfere with the protein-protein interaction, which is why we've highlighted it. And then you can see at the top here, we have the download button with all of the options. And then the API is at the top there as well. The release notes, which shows everything that's been updated recently, and the help, which shows you all of the in-depth help, which is also available via the little eyes as well. Okay, so we should be back to the presentation. Thank you. So this is something that you're privileged, I hope, to see. We haven't shown this to anywhere before. This is I would say a worldwide premiere. This is a sneak peek in what we're trying to develop next. This is ProtVar version 2. And we would love your feedback after this and help with this. But one of the things we're trying to do in the next version of ProtVar is to allow users to filter the results. So what do I mean by that? If you have a protein, but you just want variants that satisfy a particular criteria, you can use these tick boxes and sliders in order to get just the results that fit your hypothesis, for example. So examples of this would be, rather than showing all the variants, which ProtVar currently does, you could just look at the variants that have been reported before. And you can also consider variants which are PTMs, have data from mutagenesis or functional domains. You might want to only consider variants with high conservation, for example. We can also do this. And all of these criteria are combinable. We can also look at disease associations or whether the variant is rare or common. And also we can look at the structural. So variants with a predicted effect on structure. And I showed you the protein-protein interfaces and predicted pockets. That can be combined with functional data we already know, just to get the variants that fit a criteria that you care about. Perhaps you only want variants that are in interfaces. And also all of the scores we talked about. So CAD scores, alpha missense, EVE, ESM1B. We can look at just the categories that are pathogenic or benign or any combination of those. So if that, I can't see any of you, but if that excites anybody, then please contact us at protvar@ebi.ac.uk, and then I can share the link to ProtVar version 2, and then we can talk about it. You can suggest things, and perhaps you can help with a bit of testing. But it'd just be great to get some feedback from the community on whether you think these things are a good idea, what we should add, what kind of filters would be really useful to your work. Okay, so with that, I'd just like to acknowledge all the people involved in ProtVar. So 2 of them, of course, are online. Tanushree very helpfully has been helping me understand whether I'm on the right page or not, which is great. Thank you, Tanushree. And then Prabhat's here as well, who's been developing all of the amazing things that you've seen. But also the predictions that are on screen here, they were mainly done by David Burke, Jürgen Yanis, and Pedro Beltrao as part of the Open Targets project. These guys have now moved away. They're in Zurich and King's. And then our funding, Embolus funded this, and so has Open Targets. We also have funding from the UK Human Functional Genomics Initiative. And I've put the QR code there again, just in case you missed it, and also the link. So please look at it, look at ProtVar, tell us what we can do better, tell us about your use cases and how you use ProtVar, and that will help us to improve it for the community in the future. Okay, so that's the end of this particular webinar. There's more webinars coming up soon, which I think will be interest to lots of people here. The next one being accessing UniProt programmatically. That's coming up this month, 20th of November. And actually, if you use this link, you can see all of the UniProt related webinars. You can also look at the pre-recorded webinars from before. And with that, I'd like to take some questions, please.
Thank you very much, James, for your presentation. And we have had quite a few questions. Some of them have been already answered by Tanushree and Prabhat. Thank you, Tanushree and Prabhat, for that. But we still have some questions open. So I'll read those. So the very first one is actually a comment. Maybe if you can add a comment on that one. So it says, no variant is pathogenic per se. It can be classified as pathogenic in the specific context of a specific disease. Pathogenicity is not predictable. One can be predicted a loss of function, change in the 3D structure, and many more, but not pathogenicity.
Yep. Yeah, okay, so I'd like to comment on that. So this is one of the reasons that ProtVar does not offer a prediction of pathogenicity. We don't have a ProtVar benign pathogenic. What we do is display what other people have done. So the scores, the effect on structure, and then previously conducted experiments, and then it's up to the user to bring together the information that ProtVar gives you, and then make your own prediction based on those. Because as you say, it may not be, even though all the scores might be pathogenic, it doesn't mean that it is pathogenic. And even though it's a large structural change, it also doesn't mean it. And we never make that claim in ProtVar. We just bring all the information to the user to allow them to make their own decisions.
Okay. I think Tanushree has a comment.
I just wanted to add that, which is why ProtVar specifically says contextualizing human missense variation, which is a very big part of kind of making that point, is that ProtVar doesn't provide any pathogenicity predictions per se. Yeah.
Great, thank you. Okay, so next one is about AlphaFill. Can AlphaFill be used with a set of custom ligands or leads list?
No, so all AlphaFill does is get experimental structures which have ligands co-crystallized with the protein, and then align them with the AlphaFold structure. So it's not our tool. We simply lift the structures from AlphaFill, and actually they're very approximate. I said that a couple of times that it was approximate. So these are not being minimized. They are not in the perfect location for the AlphaFold structure. They're just in the approximate location according to the experimental structure. So no, I mean, it's worth checking with the people that wrote AlphaFill, but we don't use it for that. We simply take the pre-computed structures and then add them to ProtVar.
Great, thanks. Can ProtVar be used to visualize a microbial metabolite ligand binding to human cell receptor to identify the exact location it it binds to the human receptor?
That's a wonderful idea. That the host pathogen interactions are fascinating. It's not something we do yet. We have looked at a handful of these as far as doing the predictions, but not very many. It'd be great to be able to move into that direction in the future. So whoever gave that question, please feel free to email me, either my personal email or to protvar@ebi.scdk, and we can look at that together.
Great. Is information given only for known variations? What happens if it is a somatic unknown variant? Will it have the structural and some kind of score too?
Yeah. Yes, is the answer. So we have information on every possible change in every single protein, every position. So it doesn't matter whether it's been seen before in Nomad or Cosmic or any other resource. If it's never been seen before, it's entirely novel. We will have the position in the structure, which we can show you. We can predict whether it's in an interface, whether it's in a pocket, what the structural stability changes. So all of that information we can give you, whether it's been seen anywhere else or not. If it's entirely novel, ProtVar will still have it. Yeah.
Okay. I think there was a follow-up question on that one. What about variant of unknown significance?
Yeah, sure. Again, that's just a category. As I said before, we don't know when things are pathogenic or benign or unknown. This is just, helping us to simplify the interpretation from, and we don't provide any of that. So most variants of unknown significance. So what we try to provide is the contextualization so that you can look at a VUS and shift it to the more likely pathogenic or more likely benign based on all the information that we can provide you. But it's really up to you to make your own decisions based on that information.
Okay. Can you comment on using ProtVar to look into variations between species, or is it only intended for human variants?
Yeah, it's currently intended for humans. It's very important and very, very useful. The only way we use non-humans is to calculate conservation. So our conservation is based on alignments of other species as well as humans. So that's the only time that we use non-human information for ProtVar. Certainly other information is important. In fact, the experimental structures from other species are often very relevant to humans, and it's something we might add in the future. And also, it'd be great to move ProtVar into different species in the future. So again, if that person wants to let me know what their favorite species are and why they think it'd be useful to have that, then we can definitely talk about it.
Great. Okay, so there are no more open questions, but there was one question which was answered in the Q&A, but I think Tanushree has pointed out if you could comment on that one. So the question is about how is this different from CAD annotations?
Sure. So CAD is a score. So CAD and alpha missense, more recent PopEve and ESM1, they're all scores of predicted pathogenicity. So what CAD will give you is this likely to be deleterious or not, or unknown. So it'll give you a category. What ProtVar tries to show is how it's likely to be deleterious. This helps you understanding the mechanism, and it also helps you understand whether the score is likely to be true or not. It's not just a binary yes, no answer. It brings all the information we know about that region, the protein, and about the position to help you understand the variant, the how of the variant, rather than just whether it is or isn't, whether it's a 0 or 1. That's what ProtVar aims to do.
Great. Thank you. There are no more open questions at the moment, but we still have some time if anybody wants to pop in any question. There's just one comment. Thanks. Great demo of your powerful platform. So yes, as James mentioned, I think please write directly to ProtVar if you would like to participate in the testing the platform.
Please do. Yeah, please do. You can help to shape how ProtVar 2 looks. I mean, there's whatever 50 people here. You're all in the position where you can help to guide it towards your kind of research questions. So please do get in contact with us. We really need user feedback and community feedback to help serve the community the best.